I'm going to talk to you about gonadotrophin preparations we use and the evolution of the different gonadotrophin therapies. If we look at the ovarian induction or ovarian stimulation, we hardly talk about the earlier stages of follicular development from the primordial follicle to the preantral follicle. But once the follicle gets recruited from the preantral onwards for the pre-ovulatory and the process of ovulation, which is the whole process of about 17 to 20 days of follicular growth. And that's the follicular growth we are talking about. And gonadotrophins are essentially used for ovulation induction or ovarian stimulation for this particular period of follicular growth. And our whole science of IVF and reproductive medicine revolves around these days of follicular growth. The evolution of gonadotrophins has been neatly published by Bruno Lilienfeld in two of his articles, and this is the latest one of it, published in the Frontiers of Endocrinology in July 2019. And as we all very well aware about, that it was the animal cadaveric gonadotrophin which was used in the ancient times, and then by human pituitary extracts, and then the human menopausal gonadotrophins, and now we use recombinant FSH. And in this article of historical perspective, Bruno Lilienfeld has given the evolution of the gonadotrophin what we use. And initially, it was called as prolan A and prolan B, and proles means descendants. So it was basically compared to the pituitary, and there's some descendant hormones from the pituitary acting on this ovary as is depicted in the picture, and a follicular growth will take place. And then the standardization of the gonadotrophin was in question. And initially it was standardized as the rat unit. The rat unit is the one is the amount that is produced for and is responsible for forming the vaginal opening and estrus once injected into an immature rat. By 1941, a two-step protocol was formalized by saying that the follicular growth stimulation with sheep or pregnant mare serum gonadotrophin was used, and an ovulation trigger was given by HCG. And HCG was one of the first gonadotrophin to be synthesized then. Park Davis Company in 1941 introduced a molecule called cyanopoidine, and this was a combination of a low dose of HCG and pituitary extract, but then this was given continuously as a gonadotrophin for ovarian stimulation. And when it was used in those days, prolonged amenorrhea patient with polycystic ovary, a few of them did develop follicles, a few of them got menstruations, and a very few live births were also noted. But they did notice plenty of OHSS for the first time with larger ovaries, fragile ovaries, and intraperitoneal bleedings. Later on came the evolution of urinary HMGs or urinary gonadotrophins. In 1960 and 1971, mainly the urine was collected from the Italian nunneries and in the Netherlands. But as more and more gynecologists in the world started using gonadotrophin, the demand did increase. And then the urine collection for extraction of human menopausal gonadotrophin started and expanded to Spain, Brazil, Argentina, and China. And then, as you know, they are the main source of raw material even today. Urine was pooled, so individual donor details were just not available to us. Just a health questionnaire was submitted to every donor, and the donor will fill up that questionnaire, but that questionnaire was never available to the term end user, and that is the gynecologist. Unlike the blood collections, urine collection, there's no SOPs available for urine collection. So it was just a pooled urine of the nunnery which was used. And even the transportation of the urine from one part to the laboratory, from the, let's say, from the nunnery to the laboratory, was not even monitored. So there were a lot of lacunae in the whole process of urine collection and process of purification. As you are seeing in the picture, it is the urine collection in a nunnery which is going on from the can to another can. And on the right side, you are seeing a kaolin cake. So this urine will be first filtered under pressure through a kaolin cake, and then it is acidified and acetic acid is added, and further precipitation and further things will take place. And that was a very raw pictures of the HMG purification which was then done. Cyrano introduced a molecule called Personal 25, and it was the first HMG preparation registered in Italy in 1950. Definition of one unit was based on the capacity of the product to induce estrus in a 28-day-old rat. Then, Sirono donated about 50 gram of Personal 23, which was the first international reference standard against which all the newer preparations of HMG had to be compared to know the potency of that particular molecule. Those days, it was bioactivity of FSH, and Steelman Pohe bioassay was the one which was used for all those HMG preparations. In the Steelman Pohe bioassays, there were 21 to 22 day old female rats. They were randomized to receive the drug and those who wouldn't receive the drug. They were injected with the FSH for a period of 3 days, and on day 4, the rat was sacrificed. And the ovarian weight of the injected and the non-injected rat was measured, and the difference in the A weight was the one which is a bio, which proved to be the bioactivity of the FSH given to it. In 1972, in Geneva, a meeting was convened under the leadership of Bruno Lunenfeld, and certain treatment guidelines were developed from the use of gonadotrophins. And they said that an effective daily dose of gonadotrophin for hypogonadism patient was about 150 to 225 IU, and that for ovulation induction was 75 to 150 IU. And this WHO introduced this guideline for the first time in 1973. Later on, the improvements in the purification technique separated the FSH molecule. And as the purification goes on and more and more FSH is purified by different methods of chromatography, the LH activity in the final product is very low. And to maintain a good LH activity, it was replaced by HCG. Later, monoclonal antibodies were used to separate the FSH. And the use of monoclonal antibodies and the chromatography done later that resulted in highly purified FSH, and that was about 9,000 IU per gram, and that was Metrodin, and which had about less than 0.1% of LH and less than 5% of impure proteins. And this also made the injection volume smaller, and these smaller injection volumes could be given even subcutaneously. So if we look at this is the whole evolution of gonadotrophins, which we have witnessed over the last several decades. And by about 2003, from 1995 to 2003, were various studies on recombinant FSH, and 2003 onwards, recombinant FSH was made available, and now even the Quadritropin Alpha is available in Western world, which is a long-acting drug. FSH probably it should be available to us soon. From the side effect point of view, it was initially there were local reactions and a lot of side effects, and even they called it an anti-hormone because a lot of antibodies developed and which eventually decreased the pituitary function. There also origin of a disease called uh, Creutzfeldt-Jacobs disease, and this particular disease was mainly seen with the use of gonadotrophins, which were governmental preparations. The company preparations did not have peculiarly this particular disease. It was more seen in New Zealand and Australia. But later on, all these problems of uh, side effects came down, and more safe and efficacious products are now available to us. If you look at the structure of FSH molecule, it has carboxy terminals and sialic acid residues are attached to it. Depending upon how much sialic acid is attached to an FSH molecule, there are different FSH isoforms. Sadly, in commercial practice, we have only, or the commercial preparation of FSH have certain fixed sialic acid ratios. But when it comes to a natural FSH, different isoforms are secreted in different parts of menstrual cycle. If you look at this diagram, you see the FSH molecule with highly acidic, more acidic FSH molecule is secreted in the early part of the follicular phase. And this highly acidic molecule could be less biopotent, but it has a long acting. So it pushes the herd of follicles better, this long acting molecule, by maintaining and reaching the FSH threshold and maintaining that FSH threshold. But when it comes to the late follicular phase or around the luteal surge, it is a little more basic FSH isoform. There's nothing like basic actually, it is just less acidic. FSH isoform is secreted, which will have a very high bioactivity, but it will be short acting. When you see those diagrams of physiology, you know when the LH surge is taking place, there's a small nick in the FSH. That nick in the FSH is a highly biopotent FSH and short acting. Because it's highly biopotent, it recruits certain follicles for the next menstrual cycle. And again, in the late luteal phase, when the luteofollicular transition is taking place, an acidic FSH isoform is secreted in a natural menstrual cycle, which will again push the growth of the follicles further. And FSH acidic isoforms are secreted at an older age. So menopausal women will have more amount of acidic isoforms, which are long acting and less biopotent compared to the younger population, which has mainly basic forms. And acidic forms are also found more seen in premature ovarian failures and patients with poor ovarian reserve, and they do have raised FSH levels as well. So acidic forms are long acting and less biopotent, and basic forms are more biopotent because they have a higher binding affinity to the FSH receptor and a greater efficiency. Look at the diagram on the left side. When you use a very small dose and the least acidic molecule, uh, in that you see the red line, which has the highest bioactivity. And when it comes to a higher dose, then probably the acidic and basic isoforms of molecules, they simply act well. And this is some information which needs to be put into clinical practice. That if you're using a lower dose of gonadotrophins, it's better to use more amount of your basic forms because they are more biopotent and they will have higher affinity. Even at low dose, they will work better. But if you're using a larger doses, let's say you're using 225, 300 IU, it wouldn't make much difference what kind of preparations you use. And that's what is reflected in various studies. And also the ESHRE guideline on ovarian stimulation talks about you can use any kind of gonadotrophin in process of IVF. The commercially available FSH preparations are more acidic. These changes take place in the purification process, hence they have a longer half-life and they can accumulate very easily. And because they accumulate, a certain level of FSH is reached very fast and it will be maintained, or FSH could go on mounting and it will push a large herd of follicles than the natural FSH. Then came the development or invention of a polytropin alpha, that's gonal-F, and polytropin beta. And it was basically a recombinant FSH molecule. The polytropin alpha gonal-F came from the Chinese hamster ovary, and it was transfected with separate expression vectors for alpha and beta chain. And the both molecules differed in their purification process because there was bioreactor, ultrafiltration, and 5 chromatographic steps and immunoaffinity for chromatography in FSH alpha, while purification process with series of chromatographic steps and anion cation exchange and hydrophobic interaction and size exclusion chromatography was then there in the development of Recagon. Owing to the differences in the production and purification processes, both alpha and beta, that's gonal-F and Recagon, differ in the glycosylation and the sialic acid residues. Gonal-F is more acidic than Recagon, and the metabolic clearance of Recagon is about 40 hours, and that of Gonal-F is about 37 hours. They were initially, when they were introduced, it was distributed as bioassay, but then because of their consistency of mass of protein could be fixed for the bioactivity, and nowadays a filled by mass technology is available, and now the pens, friendly pens are also available. There is no difference in the clinical studies between the two drugs. The oocyte numbers, embryos, and the pregnancy rates were almost the same. So just a recap of what we studied till today, till now, is FSH is highly heterogeneous protein, and because of different sialylated links and the oligosaccharides, which cause protein to be charged negatively, and the higher the sialic acid, more acidic the FSH isoform is. Acidic FSH isoforms are more stable, they have a longer half-life than the basic isoforms, but the basic isoforms are more potent. More basic FSH isoforms have a higher binding affinity to FSH receptor and have been associated with greater efficiency than the acidic isoform in promoting granulosa cell proliferation and rapid preanterior follicular growth in vivo. During the early follicular phase, as well as the late luteal phase, the menstrual cycle, acidic forms are predominant, while near the LH surge, the basic isoforms are there. And towards the end of the woman's reproductive lifespan, acidic FSH isoform increase, whereas the basic isoforms will decrease then. And urinary FSH is more acidic than recombinant because comes from the menopausal urine, so less biopotent and higher purification process. FSH molecules will slowly move towards a recombinant type, that they will become more basic and their potency will keep on increasing. And salic acid content is different in different recombinant preparations. I think this is a very important diagram which is telling you about the purity. That FSH, you can see a band of FSH there, which is the recombinant FSH, and you look at the band of the FSH part of the HMGHP, that's Menopur molecule. They are almost at the same time. So HMGHP has an FSH which is more purified compared to a routine HMG which is available in the Indian market, and Gonalef or Recagon have the highest purification of the FSH. But there is not much great difference between the purity of the FSH molecule between the Gonalef and the HMG-HP, but there is a large difference between the routine HMGs and the HMG-HP of the Gonalef, that of FSH. And on top of that, an HMG-HP has an HCG to it, a small amount of HCG, as giving LH activity, which is completely devoid in Gonalef or Recagon. So HMGHP available today uses HCG of pituitary origin, and LH has a short half-life, while the HCG has a good half-life of 24 hours. And Mediofert is another preparation which uses HCG from pregnant women of urine, which is not yet available to us. And Mediofert has a shorter stimulation, more oocytes are there, equal pregnancy rates, and higher estradiol levels. So we really don't know if Mediofert will cause more amount of OHSS in our population. The main difference between an HCG driven and a recombinant LH activity of the LH is an HCG driven LH has a longer half life, higher potency, better oocyte quality, and a better embryo quality, while the recombinant LH has a short half life, it's costly, and double injections may be needed in many stimulation protocols. So, a first available molecule of HMG, which was purified by hydrophobic interaction chromatography and anion exchange. And the LH molecules were preferentially lost here because you wanted to purify the FSH. And the bioactivity of LH will go down. And hence, in Menopur HMGHP, the bioactivity of LH is added by HCG, and about 9.9 MIU of HCG corresponding to 70 to 75 IU of LH is there. There were systematic reviews which are published comparing HMG, HP, and recombinant FSH by Alinani in 2008 and 2009, and that by Kurosami et al. In 2008. They attributed the superiority of HMG, HP to FSH, and it was attributed to the presence of HCG and LH activity. And more than two-thirds of the patient in the HMG arm of all these systematic reviews were using Menopur as their HMG. So HCG acts on the LH receptor, and HCG binds to LH receptor, which is immobile, while LH binds to an LH receptor, which is rotationally mobile. Hence, there is a different receptor activation. HCG has a five-fold higher affinity to LH receptor than LH, and HCG is more steroidogenic, cyclic AMP stimulated. It supports the early pregnancy better, and better progesterone release is also there. And LH has an anti-apoptotic ARK1 and ARK2 pathway, so the oocyte maturation is more seen better with LH activity there. So when it is RFSH, it's only a basic FSH, recombinant FSH. HMG will be FSH, will be acidic, and there will be an LH of pituitary origin. While in Menopur, which is HMGHP, FSH is more basic compared to a routine HMG, and due to higher purification process, and HCG, which is a more potent LH activity, is there with it. And what happens with an HMG response? What's the difference between an HMG response and a recombinant FSH responses? With HMG, you would get less number of follicles, but the maturity of the follicle will be better because the two-cell, two-gonadotrophin theory is better acting there because of a good LH activity. Less amount of OHSS will be there. Less progesterone rise on the day of HCG compared to RFSH. So a fresh embryo transfer is possible. More live birth rate and cumulative live birth rate have been reported with trials with less miscarriage also reported in all the trials published till date. Menopur, this is about batch to batch variability. The Menopur contains both LH and HCG molecules, but LH is lost. More than about a lakh of individually screened donors contribute to the urine several times, and this ensures a consistent starting material, and variation in the batch is quite minimized. So at the raw material point itself, an HMG HP they start devising the batch properly. Anion exchange chromatography purification maintains a consistent sialic acid content, thus affecting the bioavailability, and hence the batch to batch variability of an HMGHP like Menopur is not is much less and is not like the HMGs which of the other simple HMGs which are available in the market. Now, this paper I have particularly included because I see a lot of consultants using a recombinant FSH in the initial part of the stimulation, followed by a urinary HMG, which will obviously have a urinary FSH. But whenever those kinds of studies are done, they have seen not much difference in the pregnancy rates in the treatment arm and in the arm where a preparation was used consistently. But this particular study, published in Gynecological Endocrinology in 2014, used a urinary FSH in the initial part and the recombinant FSH in the later part. So the urinary FSH having more acidic form with a longer half-life and more basic form with a shorter half-life and high potency was used in the later part. And then they noticed an improvisation in the embryo grading. You look at the lower table, which says about 50.1% of group A had a embryo grade 1 compared to 38.8%, and pregnancy rates are also better with a p-value of 0.05, and implantation rate was good. So it is not practical, it's not advisable to use recombinant FSH in the first part followed by HMG. You can do a reverse or use just one preparation consistently through the whole stimulation protocol. This is a well-known diagram which talks about the development of LH receptors. LH receptors generally are very few in the follicle is about 2 to 6 millimeter. By 10 to 12 millimeter, about 50% of the receptors are LH receptors. 50% granulosa cells will have LH receptors. But by the time the follicle starts growing beyond 14 millimeter, about most of the LH receptors will be there at that time. So this particular follicle beyond 14 millimeter is less dependent on FSH and is more dependent on LH for its maturity, estrogen level, which ensure a good endometrial development and a good hormonal profile of the luteal phase as well. So this is one interesting study which is done by Felikori et al. And published in 2003, if I'm not mistaken. It's about giving a certain amount of LH activity in the late follicular phase. And what they did was in the initial 7 days, they gave recombinant FSH, and followed by that, they gave a certain amounts of HCG. Like in one group, it was the recombinant FSH, which was continued. In another group, they gave 50 IU of HCG, another group 100 IU, 150 IU, and 200 IU. And what they found is LH will act like an androgen substrate, but not only that, this HCG can directly stimulate the granulosa cell stimulation will be there and a better maturity would be there. It will continue the follicular growth with decreased FSH in the late follicular phase. So in the late follicular phase, first FSH just push the follicle to 14 and now giving a good amount of LH activity will maintain the growth further. And with increased FSH levels, LH and HCG support later on and larger follicle growth will be there. But if the initial FSH itself, whatever you're given, has not given an adequate growth in the follicle, replacing that with HCG later may not do well. So you need to have a good growth initially, then only you can have that HCG acting better. A decrease in FSH with LH replacement hastens the demise of smaller follicles, and it is the VEGF and the hormone which is secreted from the smaller follicles, which also contributes to OHSS. And decrease in the amount of smaller follicles is very beneficial from prevention of OHSS. And more large follicles and less small follicles, it's pretty similar to a natural menstrual cycle, and getting better pregnancy and better mature eggs is good. This is a same slide about the half-life of HCG and LH. And look at this particular thing. And these are the studies which are published in 2012 by Thusin et al. They talked about the top quality embryos when an HCG supplementation, as Felikori did. Because that Felikori study in 2003 was about IUI cycles. And this is about the IVF. And then they saw that whenever they gave HCG in about 50, 100, 150 IU, they obviously had more top quality embryos whenever HCG supplementation there, and the live birth rate, cumulative live birth rate was better. So Menopur and HMGHP allows you to use this HCG in combination with a highly purified FSH, decreasing the growth of the smaller follicles, giving more larger follicles, and obviously adding to the top quality embryos. And when we talk of top quality embryos today, we are talking about the cryo survival because a lot of our cycles are freeze all cycles. And if you look at this particular study about whether to use HMG-HP or recombinant FSH on the embryo quality parameters, and they really went through the studies published by the Merit trials, and they found that the HMG-HP, the gray bars are of HMG-HP, they had more blastomere survival rate compared to the recombinant FSH. And when it comes to cumulative live birth rate or live birth out of the first cycle, obviously the HMG-HP had more cryo survival and more live birth were noticed with that. So when it comes to a cryo survival, and probably that's the crux of an embryo, that we want an embryo to do very well. You want all the blastomeres to survive well, and probably HMG-HP will work better there. Coming to the different recombinant FSH, Polytropine alpha is more acidic than Polytropine beta, that's Recagon. Beta is more potent than alpha. So the clinical pregnancy rate is better still with alpha because that is gonal compared to Recagon, because with Recagon there are higher E2 levels that could be changing the endometrial receptivity, and that's been very well published by Rola Orivito in 2009. What about Quadritropin Alpha? Is not yet available to us. It's a long-acting molecule, and they borrowed a carboxy terminal of the beta chain of HCG, and that added a long half-life. So newer protocols which will come as this drug comes to us probably could be one dose of Quadritropin Alpha, and after 7 days we'll be adding an antagonist and a daily dose of FSH or daily dose of HMG to our protocols. And the clinical studies which have been published are saying that the live birth rate, ongoing pregnancy rate, and clinical pregnancy rate remains the same. But if you use a lower dose of polytropin alpha, your live birth rate may suffer. There's something called Polytropin delta, which is not yet available to us. It is from the fetal retinal cell line, and Polytropin epsilon from the human blood myeloid leukemia cell line. It is not yet marketed to us, and probably the future of gonadotrophins could be here. And we could see many more other things also coming to us, and that is one of them is oral antagonists like elagolix. And we also have some fixed drug combination preparations, like we have a recombinant FSH and recombinant LH mixed together available to us, and it is, and even the HCG is recombinant HCG like Ovitrel is now available to us in a pen format, which can be given in low dose either in the late follicular phase, or it can be used as a luteal support by giving low dose on a continuous basis by the various studies, as per the various studies published by Peter Humaden et al, wherein they give about 125 IU of HCG, urinary HCG, every other day as a form of luteal support. So practically, the take home would be if you're using a low dose, please use recombinant FSH and Menopur because they're highly pure drugs. They have a good FSH activity, and obviously your stimulations will be better. And here again, I would like to say that in PCOS women, try and use more and more of HMG HPs because that's where your HMG HP will avoid OHSS and give a better quality of embryo, better estrogen level, and the pregnancy rates will be more as been devised with the Megaset trial as well. And if you're using a high dose, obviously there are various options and you go by the economy. And as the HRA guideline also says, you can use various drugs. But again, I would say when it comes to a pregnancy rate, especially in low ovarian reserve patient, they do require a higher push with a good FSH activity and a good LH activity as well. So in that group also, HMG HP with a good amount of FSH and HCG is a good drug. Or you will have to use a recombinant FSH with an added HMG so that you have, or a recombinant LH there to push on the follicular growth in patients of diminished ovarian reserve. Well, thank you so much. And I sincerely appreciate uh, for your patient listening. And thank you again.